NASA has begun a crucial experiment to see how bacteria such as E. coli could affect astronauts. The space agency shot a sample of the bug into space from the International Space Station. They believe that in zero gravity, the bug could have an increased resistance to antibiotics, which could cause major problems for astronauts. Blast off. After arriving at the International Space Station, crew worked with ground controllers to release the satellite into orbit, and it is programmed to automatically begin its experiment. The experiment: if we find resistance is higher in microgravity, we can do something, because we'll know the gene responsible for it, and be able to design countermeasures, said A. A.C.Matin, principal investigator for the EcAMSat investigation at Stanford University in California. If we are serious about the exploration of space, we need to know how human vital systems are influenced by microgravity. Scientists believe that bacteria like E. coli may experience stress in microgravity. This stress triggers defense systems in the bacteria, making it harder for antibiotics to work against them. Bacteria on Earth do something similar by developing a natural resistance to traditional antibiotic treatments. By knowing how E. coli's resistance to antibiotics changes in space, we can also better understand bacteria on Earth, leading to more effective treatments here too. The E. coli strains used on EcAMSat are responsible for urinary tract infections, which can happen to astronauts in space in addition to other types of infections. Scientists hope the results will help them work the ideal dosage of medicine to combat E. coli infections in space, and explore other techniques that could enhance the power of antibiotics that already exist today. EcAMSat is a uniquely autonomous satellite, meaning it can conduct its experiment without any communication from Earth. After arriving at the International Space Station, crew will work with ground controllers to release the satellite into orbit, and it is programmed to automatically begin its experiment. This CubeSat, a spacecraft the size of a shoebox built from cube-shaped units, has just been deployed from the space station. Beyond low Earth orbit, the compounding human health effects of microgravity and space radiation will require more knowledge about how biology reacts to the space environment, said Stevan Spremo, project manager for the mission at NASA's Ames Research Center in California's Silicon Valley. Lessons learned in this experiment will serve as a stepping stone for more advanced biological CubeSat missions, answering critical questions. Though EcAMSat will only fly this once, many of its components may embark on a different mission. Life detection in the solar system, said Tony Ricco, chief technologist for the mission at Ames. How the experiment will work. EcAMSat is a uniquely autonomous satellite, meaning it can conduct its experiment without any communication from Earth. After arriving at the International Space Station, crew worked with ground controllers to release the satellite into orbit, and it is programmed to automatically begin its experiment. Students at Santa Clara University in California will monitor the spacecraft, handle mission operations and download data. The spacecraft will awaken the dormant E.Coli by flooding them with a nutrient-rich fluid, adjusting the containers to the temperature of the human body, and then injecting the bacterial samples with different amounts of antibiotics. Two types of E.Coli will be compared, one with a naturally occurring gene that helps it resist antibiotics, the other without. The experiment, a spacecraft the size of a shoebox built from cube-shaped units, has just been deployed from the space station. The bacteria will be mixed with a dye that changes from blue to pink. A dye that remains blue indicates most cells have died in reaction to the antibiotic. The more cells that remain viable and active in spite of the medicine, the stronger shade of pink the dye becomes. An onboard color sensor will detect these changes and determine how strongly the two types of E.Coli resist the antibiotic at different doses. The experiment will last for 150 hours as EcAMSat orbits the Earth, and the dataset, less than a megabyte in total, will then be transmitted via radio down to Earth. After the conclusion of its mission, this little satellite will burn up in the Earth's atmosphere some 18 months later. Using sensors and the microfluidics technology from EcAMSat, NASA is developing the technology needed to look for life on moons such as Enceladus and Europa, ocean worlds covered by icy crusts.